Chapter 13, Section 2, Chemical Logic and Common Biochemical Reactions. When we look at the biochemical reactions that are going to occur in a cell, the 5 predominant ones that we will be discussing throughout this semester are listed here. You see that the first one, the making or breaking of the carbon bond. Well, yeah, you're going to have to link carbons together to take small molecules and make much larger macromolecules. You're going to have to come up with some way to rearrange those molecules. My mantra for my courses have always been, shape dictates function. So, change the shape. Change where a carbonyl group is. Change where a hydroxyl is. You make those changes, you change the shape. You change the shape, you change the function. Free radical reactions are also going to be things that we see that occur within the cell. Living cells, prokaryotic, eukaryotic, doesn't matter. An atom or a molecule that is lacking an electron or has heavy electrons is going to be more reactive than those that have the valence shell full or have the valence shell half full, so they're quasi-stable. Going to have to change groups. Which groups? Functional groups. Add a sulfhydryl, add a carbonyl, add a carboxyl. Take one off. Move it from one molecule to another. And then the oxidation reduction reactions. Who has the electron? Where is it coming from? Where is it going? Now, let's look at this a little closer. When you go and you break bonds between atoms in a molecule, we know that the bonds holding 2 atoms together in a molecule, as you see here, carbon and the hydrogen, or the carbon-carbon bond, this is actually an electron pair being shared. 1 bond is 1 pair, 2 bonds, 2 pairs. When you go and you break that bond, well, means you're going to divvy up the electrons that form that bond. If you divvy them up so that they're equal, each atom that was part of that bond gets one of the electrons. We refer to this as homolytic. This homolytic cleavage is going to result in a radical. Notice it has 1 electron, 1 electron. Its valence shell is not happy. It's going to be highly reactive. If, though, the division of those electrons that were part of that bond is unequal, 1 atom, as you see here with the carbon, keeps it. Hydrogen goes away. It's just a proton. Or if you look on the slide, your handout's behind my head, or you look in the textbook at this figure, you'll see the carbon-carbon splits. 1 carbon gets both electrons, the other doesn't. Giving you a carbanion or a carbocation. But for those bonds to be broken, for things to be shifted, there's going to need to be some changes in where the electrons are. Who's got them? Where's it coming from? If an atom in a molecule is readily available to give up an electron, we refer to this as being a nucleophile. It will donate the electron. It's going to donate the electron. So that a bond can be formed. If an atom that is part of a molecule is readily available to accept an electron, means it is an electrophile. It's going to want to interact with, bring in the electron. Here you look at this carbonyl group that's part of a much larger molecule. The R is some other group, some other molecule, some other atom. The red arrows denote electrons being passed. So R here is going to be the nucleophile, C is going to be the electrophile, as it takes and accepts the electron from R. There's going to be a shift, so R is now going to be bonding to C, so there's going to be a bond here, but that would give carbon too many bonds in its valence shell. So this double bond, the carbonyl, the double bond for the carbonyl here, oxygen is going to take one of those electron pairs, so it'll be a carbon singlet oxygen. Chances are a hydrogen is going to have to be stripped from somewhere and brought in and turn this carbonyl into a hydroxyl group. As I mentioned a few slides ago, the heterolytic breakage of a carbon-carbon bond doesn't result in an equal distribution. It results in an unequal distribution, so that you end up with a carbanion, which is, well, I'm trying to draw here, one, that's supposed to be two dots, that's carbon negative, and then C that is plus because it is light electrons, heavy protons. Carbanion, carbocation, they are going to react differently in the cell. What we see here when we look at the carbonyl groups, the chemical properties of these carbonyls, is such that the carbonyl group is an electrophile. It means that the carbon here is going to be accepting an electron from someplace else. Why is it doing that? Well, it's right here in A. This is actually figure 13, was it 3 from the textbook. The carbonyl group has an unequal sharing of the electrons. Oxygen's a greedy bugger. It's going to hold on to the electrons longer than the carbon of this carbonyl group. So it means oxygen is going to have a negative-ish charge. Carbon's going to have a positive-ish charge. Kind of like what we discussed last semester with water. The hydrogen and the oxygen. Here we see the same thing though, but it's going to be across this double bond between oxygen and carbon. Here with B, we see that this neighboring carbon is going to do a nucleophilic attack against this carbon of the carbonyl group. That carbon's acting as an electrophilic carbon, so it's going to readily accept. And we see bonds shifting. So this electron goes up. This oxygen that was balanced now becomes negatively charged, polar. That open electron here now goes from this carbon to the neighboring carbon, and you get a carbon-carbon double bond. We can see the same thing if you have, instead of an oxygen, you have a neighboring amine group. We can see the shifting of the bonds. The double bond here between the carbon and the nitrogen shifts, and now you have the nucleophilic attack against this electrophilic carbon, and you get the shifting of the bond from carbon to nitrogen to now a carbon-carbon double bond here. When we look at the different condensation reactions here are going to be the joining of different molecules. Here we see nucleophilic attack to an electrophilic here carbon from this carbon, and the rearranging of the bonds. The carbonyl groups, C double bond O, now becomes a hydroxyl group as that electron shifts and a hydrogen is scrounged from somewhere. We now have go from a 2-carbon to a 3-carbon chain with a terminal hydroxyl. We see roughly the same thing occurring here when we're talking a Claisen condensation. But instead of a carbonyl group becoming a hydroxyl, we see the loss of the bond between the carbon and a sulfur group. The release of that sulfhydryl, whatever R2 is, and the sulfur, that's released. And we now have a terminal carbonyl group. These reactions can be utilized for the making of larger and larger molecules. A little hard to see here in this video because my big old head is covering. But if you go and you look at figure 13.5 or you look at slide 8 in the handouts for this video, you'll see that if you take the compound dimethylallyl pyrophosphate, cool, dimethylallyl phosphate, and I'm probably mispronouncing that. You go through and you utilize nucleophilic attack from this carbon terminal methyl here against the carbon terminal carbon here. In the process, you're releasing the 2 terminal phosphates here on diethylallyl pyrophosphate. You're bringing in isopentyl pyrophosphate, which if you look is quite reminiscent, but the difference is right here, diethyl has this carbon-carbon double bond, which is lacking here. It's actually a terminal carbon-carbon double bond, which is going to be good because nucleophilic attack from this carbon to that carbon, this carbon towards that carbon of the incoming chain from diethylyl. It's no longer pyrophosphate because the 2 phosphate groups have been removed. And what you can end up with is a much larger carbon chain. Goes from 2, 3, 4 carbons, bringing in 1, 2, 3, 4. So now you have an 8-carbon chain. And it's all based on nucleophilic attacks of electrophile carbons. As I said at the beginning of this lecture, this video, a lot of what we see when it comes to cellular metabolism is actually based on rearranging of intramolecule functional groups, moving bonds around. Where's the carbon-carbon double bond? Has it been moved? Where's the carbonyl? Where's the hydroxyl? Have they been moved around? Just moving things within the same molecule. Remember, this is the isomerization. Shape dictates function. If the original shape has a carbonyl on carbon 2, and you move it so now on carbon 1, you went from a ketone to an aldol. Makes a big difference. Also going to see this around carbon-carbon double bonds. What was cis is now trans. What was trans is now cis. So if you want to go from a carbon-carbon single bond, need to open up some electrons. Well, need to remove water. Hydrogen, hydroxyl. 2 H's and an O. Water. Means this carbon and that carbon now have electrons in their valence shell that are ready to be shared. So you now go from 2 carbons. Remember, anytime you have a carbon in a chain, or carbon with 4 valence, 4 bonds in its shell, it's a tetrahedron. So you're going to have this weird quasi, it's supposed to be a pyramid, but I can't draw it with a mouse. And you're going to end up with, a second pyramid. So you're going to end up with 2 inverted pyramids, is what this looks like. But then you go and you build this, and what you end up with is something that looks like a flat plane. R group here, H, H, R. So, 2 inverted pyramids that are like this, versus something that's now is a plane flat like this. Shape dictates function. The isomerization reactions, like I said, have a terminal carbonyl, carbon 1, carbonyl, carbon 2, hydroxyl. Second enzymatic step in glycolysis is taking glucose 6-phosphate and turning it into fructose 6-phosphate. This is done by phosphohexane isomerase. Remember, anything in enzymes is an enzyme. Isomer means it's a isomer-making enzyme that recognizes phosphohexane. Hexane's a 6-carbon sugar. All it does, swap these 2 things. Now, it's a ketone. Carbonyl is on carbon 2. Was an aldol, now it's a ketone. This is driven by, like I said, an enzyme. That enzyme, phosphohexose isomerase, in its active site has a set of R groups on amino acids that form the active site. Here, those R groups, B1, B2. B1 and B2 interact directly with the terminal end of glucose 6-phosphate. You have an electrophilic attack from B1 to this hydrogen, and the hydrogen here on B2 acts as a nucleophile, allowing for this double bond oxygen here to interact there. So you now will have inadvertently for a while a carbon, oxygen, hydrogen, and that hydrogen was there at B2. Gets pulled away, and now you have an electrophilic attack to this hydroxyl, where it's going to strip that hydrogen away, allowing this oxygen, electrophilic attack on this carbon. So this intermediate results in this, The hydrogen of the hydroxyl for carbon 1 originally came from the B2R group, which was part of an amino acid in the active site of this enzyme. This hydrogen here gets shifted, pulled away intermediately, just for a short term by B1, and then this carbon-carbon bond, double bond here that was formed, electrophilic attack bringing that hydrogen back. Because remember, if carbon 2 has a carbonyl, double bond oxygen, it has no room for that hydrogen. That hydrogen has also been shifted. And when you look at free radicals, we're going to see part of the problem is free radicals, as you see here as this X group, part of some molecule, or it's a molecule itself, has that one electron there. It's going to be highly reactive. It's going to be highly reactive, and as you see here, it does an electrophilic attack on this hydrogen, which was going to mess with the bonds here between that hydrogen and that carbon, and that hydrogen gets stripped away, which results in nucleophilic attacks, nucleophilic attacks here, and the next thing you know, you have this part of that carbon chain released as CO2, and you go from, what is that originally? Coproporphyrinogen 3 now becomes protoporphyrin 9. Same base ring, but now a much abridged carbon chain added to it. Side note, as a biologist, geneticist, we really, cell biologists, we worry about this free radical attacks, not just on this coproporphyrinogen, but more importantly, on things like nucleotides, your DNA. Start stripping off, messing with the nitrogenous bases on your nucleotides, and yeah, before you know it, you could induce mutations. Mutations could be passed on depending upon what cells that damage occurs in and at what point in your development it occurs. Now we also have to have a mechanism for the transference of groups, those functional groups, bringing in, a sulfhydryl, bringing in an organic phosphate, bringing in different things. The example shown here, this carbon, part of something, has a carbonyl, double bond oxygen. X is some other group there. Could be, a methyl group, could be a continuation of whatever. R is the majority of whatever this molecule is. You have an electrophilic attack nucleophilic attack here by Y against this carbon, which is going to be electrophilic. It's going to say, yes, please come. The electron here is going to shift back up to the oxygen. Oxygen, during this tetrahedral intermediate, is going to take on a polarity. It's going to be negatively charged. And now you see this very, very unstable structure. This is unstable, so this electron here is going to come back. You're going to have that carbonyl reformed, and this bond is going to be shifted away. These electrons are going to be shifted away, and now you have X released as a radical. Y is now part of the molecule. Glycosyltransferases, we see the same thing here. I'm not going to go into the SN1, SN2 pathways. The important thing we're going to see, one of the important things, is the phosphate groups, as you're seeing in lab, as we're going to be talking about the kinases, the signaling enzymes, the passing around of a inorganic phosphate from ATP, GTP, CTP, UTP, TTP, whatever. Normally we draw it as it shows here in A. 3 single bond oxygens around a central phosphate and 1 double bond oxygen. The single bond oxygens are all negatively charged, thus this is polar. What A is showing us here is that we may draw it that way so that the oxygen to the right has the double bond, but we have to keep in mind that double bond kind of cycles around. Okay, it is not set in stone, that shape. When you have adenosine triphosphate, you have the third phosphates. It's the adenosine nucleoside, carbon, oxygen, phosphate, oxygen, phosphate, oxygen, phosphate, and so on. All single bonds. But the thing is, as we're going to see with the kinases, the enzymes that strip off this third phosphate and add it to their target substrate, thus transferring that negative 7.3 kilocalories per mole of delta G goodness, you're going to have some nucleophilic attack from a hydroxyl on the target. That oxygen is going to attack. It is going to allow for this oxygen here between the 2 terminal phosphates to remain. This phosphate is going to be transferring over, so it'll be the phosphate group, oxygen R, but, and it's hard to see, go look in the handouts or figure, what is it, 13.8 in the textbook. It'll be 3 oxygens from that were with the ATP. That 4th oxygen is going to be the one from the hydroxyl that's here for the glucose for this example. 3 oxygens, 1, 2, 3. This 4th one for this inorganic phosphate stays for P2, the beta phosphate. Everything transfers over. The hydrogen is just released. So this hydroxyl group on glucose, this is the nucleophile. The adenosine di-2-phosphate, that's the leaving group. That's what released, no longer used by the kinase. And when you go and you look at these different enzymes, remember, N's and A's, it's an enzyme. Whatever prefaces A's tells you what the enzyme does. Kinases strips the terminal phosphate off of ATP, transfers it to an acceptor molecule. Generally, that acceptor molecule is going to be that enzyme's substrate. Phosphorylases are going to take a phosphate from something, inorganic phosphate, or that's just free, or they're going to strip it off of a substrate and transfer it to another one. Strip it off of something not ATP. To reset whatever these two do, you have to have a phosphatase. Phosphatase goes and strips off, allows things to reset. Remember in lab, I made the comment that the kinases that we're studying for lab are like dominoes. Boom, 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 boom. They knock each other down, and as they're transferring that phosphate from ATP, well, phosphatases set those dominoes back up, reset the system. And I'll let you go through and synthases, synthetases, lysases, let you read up on what all these different enzymes do. The thing to keep in mind, many of the reactions we're going to be looking at as things get built up or things get broken down is going to be movement on this scale of oxidation, least oxidized, most oxidized. Methyl group, CH2 hydroxyl, carbonyl with, maybe something, carboxyl, and so on. Movement, the addition, the movement of things from the terminal to somewhere else. It's all going to be based on where the hydrogens are, where the electrons are, where they're coming from, where they're going. And you look at this reaction here, this oxidation reduction reaction by lactate dehydrogenase. During low oxygen levels in cells, they go through glycolysis. They've already reduced all of their NADHs, so glycolysis stops. Oxidative citric acid cycle stops. All of that stops. Well, cells will take pyruvate, turn it into lactate, free up a few NAD, so they can go through glycolysis couple more times, and keep going and going. Once the oxygen levels come back, well, you have this buildup of lactate. So the enzyme lactate dehydrogenase is going to take lactate and turn it back into pyruvate. And then that pyruvate will enter into the mitochondria, into the citric acid cycle, and carry on with oxidative phosphorylation. So lactate dehydrogenase can take pyruvate, turn it into lactate, take lactate, turn it back into pyruvate. Back and forth. The difference is right here at carbon 2. Carbonyl, hydroxyl. Hydroxyl, back to carbonyl. The oxidation reduction, you cannot have one without the other, okay? Loss, gain of electrons. Biologically, look at this, loss, gain of hydrogen. Usually hydrogen is what you're going to be seeing moving. Not all the time. But you watch it and you see the molecules. It's not going to be just a free electron moving. It can sometimes be that. But more often than not, it's going to be a hydrogen acting as an electron carrying along a proton. So it's the movement of these electrons, these high energy electrons, back and forth and around across membranes, which we're going to see in this semester is what drives energy, the making of energy in our cells. Need to keep in mind, those of you watching this course who are chemistry minded, you're going to be looking at some equations listed in lectures, some equations in the textbook, and they're not going to add up. You've spent so much time in so many chemistry courses balancing equations, making sure all the hydrogens on one side match up to all the hydrogens on the other, all the oxygens, all the carbons, one side, the other. Well, biochemical reactions, we really don't worry about that so much. Biochemical reactions, we're looking more at what's the grand scheme of things. What is happening overall? Not all the little nuts and bolts. From a biochemist standpoint, ATP plus water yields, what is it? ADP, and inorganic phosphate. Because remember, it's a hydrolysis reaction. that's going to release that terminal phosphate. Biochemical standpoint, we would accept that. Cool. Yeah, here's the reaction that's occurring. The chemists are clawing their eyes out, banging on their computer screens right now. No. Where'd the hydrogens go? Where'd the oxygens go? Yes, you can go through and add in. I mean, technically, if you're looking at this from a chemical standpoint, it should be, and it shows it in your textbook, ATP 4- plus H2O yields ADP 3- plus HPO2 -4 plus singlet hydrogen. That would be the true chemical. And a lot of the times we just, you know what, that's just too much. We don't need to know all that. We just want to know what's happening in the reaction. Generalized. This is enough.